Active transport. Active transport occurs against the concentration gradient and is mediated by carrier proteins. Metabolic energy is used to move ions or molecules against a concentration gradient. Active transport results in the accumulation of the solute on one side of membrane. Active transport is different from carrier proteins mediated facilitated diffusion. Active transport is of two types, primary active transport and secondary active transport. Primary active transport. It is also called as direct active transport, which is coupled directly with a metabolic source of energy such as ATP hydrolysis or absorption of light by a carrier protein in halobacteria. Transport of sodium and potassium by carrier protein, sodium potassium ATPase, is the most common example of primary active transport. Virtually every animal cell maintains lower sodium concentration and high potassium concentration than they are found in its surrounding. This imbalance is established and maintained by an active transport system in the plasma membrane, involves the carrier protein sodium potassium ATPase coupled with breakdown of ATP. For each molecule of ATP hydrolyzed, 2 potassium move inward and 3 sodium ions move outward. It is an example of electrogenic pump because it contributes to the charge separation across the membrane. It is the heterodimer composed of an alpha subunit and a glycoprotein beta subunit. The alpha subunit is responsible for ATP hydrolysis and ion pumping, while the beta subunit is important for the correct assembly of the ATPase in the endoplasmic reticulum. The alpha subunit contains 10 transmembrane segments. Certain steroids like digitalis and wabane, which are known as cardiotonic steroids, are also called cardiac glycosides, are potent inhibitors of the sodium potassium pump. These compounds inhibit the dephosphorylation of sodium potassium pump when applied on the extracellular phase on the membrane. These cardiotonic steroids are used in the treatment of congestive heart diseases because they increase the force of contraction of heart muscle by inhibiting the sodium potassium pump. It happens as the level of sodium increases inside the cell. This slows the sodium calcium exchanger, leading to increase in concentration of calcium inside, which is ultimately increases the forces of a contraction of the heart muscle. Pelitoxin, which is a toxic non-peptide substance, also inhibits the function of sodium potassium pump. It converts sodium potassium pump into a non-specific ion channel, which allows the passive transport of both sodium and potassium ions, thereby destroying the ion gradient. The active transport of calcium across the plasma membrane is driven by calcium pump that is structurally related to the sodium potassium pump and is similarly powered by ATP hydrolysis. The calcium pump transports calcium out of the cell, so intracellular calcium concentrations are extremely low. Secondary active transport. Secondary active transport is an indirect transport occurs when endergonic or uphill transport of one solute is coupled with the exergonic, which is downhill flow of the different solute that was originally pumped uphill by primary active transport. Secondary active transport is either symport or antiport depending on whether the two solutes move in the same or opposite direction. A common example of secondary active transport is the symport of sodium and glucose. The transmembrane protein sodium glucose transporter, also known as sodium glucose cotransporters or symporters, allows sodium and glucose to enter the cell together. Sodium glucose importers are a family of glucose transporter present on the apical surface, that is facing the lumen of the epithelium cell of the small intestine, and actively transports glucose molecules into the cell from the gut. The sodium flows down their concentration gradient while the glucose molecules are transported against their concentration gradient into the cell. Later, the sodium is pumped back out of the cell by the sodium potassium ATPase and thus maintaining the inward sodium gradient. The GLUT, confined to the basolateral or basal and lateral surface of the cell, allows the same molecules to leave the cell by facilitated diffusion into the extracellular fluid on the other side of the epithelium.